Hello. So this is finishing up chapter 10 and we'll start chapter 11. So we kind of left off in this area and I will review part of it. So we're talking about the neuromuscular junction, and the neuromuscular junction is where the nerve ending meets a muscle fiber, and the junction is the synapse in between. So skeletal cells are voluntary. So they're excited by axons, a somatic motor neuron. So axons is the long part of a neuron that carries electrical charge. So each muscle fiber is controlled by one motor neuron. A contraction begins when a motor neuron impulse stimulates an impulse in a muscle fiber, and the neuromuscular junction is a place where the motor neuron communicates with the muscle fiber. So I'll go into this in more detail. You don't need to know about the physiology of it too much yet. So again, the components of the neuromuscular junction are the synaptic knob, which is actually the expanded tip of a neuron axon. Synaptic vesicles, so vesicles, just like in endocytosis and exocrine glands and such, that it's in a membrane sac filled with acetylcholine, which is a neurotransmitter. The synaptic cleft is the space separating the knob and the motor end plate, so the space between the neuron, the axon terminal of the neuron, to the muscle fiber. The motor end plate is the beginning of the muscle fiber of the sarcolemma with many folds, and it's under the synaptic knob. And then there's acetylcholine receptors and acetylcholinesterase, which is in the synaptic cleft, and it breaks down acetylcholine to prevent continuous stimulation. Otherwise, you would get muscle cramping or constant contraction of that muscle, which is not fun. It's very painful. So again, let's just go over the neuromuscular junction. See here, as I said before, one neuron attaches to one muscle fiber. So here's one neuron attaching to one muscle fiber. One neuron, one muscle fiber. So the space in between here, the bottom of this neuron, and the superficial side of this muscle fiber, that space in between, they don't actually touch, is called the synaptic cleft, or the neuromuscular junction in this part. So this is the neuron coming in, so this is part of the nerve impulse. There's the synaptic knob, right? And the endomysium is right here. And the synaptic knob, motor end plate. So this is a coronal slice, synaptic vesicles, and sarcolemma. So this is what it looks like on the inside. So physiology of the muscle contraction. So you just need to know these steps. You don't really need to understand them quite yet. But if you would like to know more about them, you can feel free to ask me anytime. So a nerve impulse, so something from the neuron, causes vesicles to release to acetylcholine into synaptic cleft. So that impulse causes the neuron to put out neurotransmitters, which are acetylcholine, into the space in the neuromuscular junction of the cell, muscle fiber and nerve cell. So acetylcholine binds to receptors on the motor end plate. It initiates a muscle fiber impulse. Acetylcholine is not bound to receptors, quickly broken down by acetylcholinesterase. And this is to prevent continuous contraction, right? So we need a neurotransmitter, something that breaks down a neurotransmitter. So spread down the impulse down the T-tubules causes calcium to leak out of the terminal cisternae and into the sarcoplasm. Sarcoplasm is in the sarcomere. So here's a picture of that. Here's the neuromuscular junction. See, these neurotransmitters are acetylcholine right here. Acetylcholine binds to the end of the synaptic terminal, fuses, and releases the neurotransmitter. So these are receptors for acetylcholine, and this is the acetylcholine binding. This is the motor end plate, right? So this is the end of a muscle fiber, hence the end plate. Here's a muscle impulse, sarcolemma. It's just a thin layer on the outside of the muscle fiber, the myofibril, right? So this is going to be made up of microfilaments, and then this impulse goes into the sarcomere right here. So this is a sarcoplasmic reticulum. Remember, it kind of looks like the endoplasmic reticulum. And these cells stimulate in here, and this causes a calcium release. So this is the calcium release right here. So as it says here, nerve impulse triggers release from acetylcholine from the synaptic knob into the synaptic cleft. Acetylcholine binds to acetylcholine receptors in the motor end plate of the neuromuscular junction and initiating a muscle impulse in the sarcolemma of the muscle fiber. Part 2 is as the muscle impulse spreads quickly from the sarcolemma along two tubules, calcium ions are released from terminal cisternae into the sarcoplasm. So as we talked about earlier, calcium has many functions, and here it helps with muscle contraction, as well as there are voltage-gated calcium channels here that cause this neurotransmitter to be released from the presynaptic neuron. So calcium ions bind to troponin, and troponin changes shape. So troponin moves tropomyosin, exposing active sites on actin. Myosin heads bind to active sites and form cross bridges. Remember, myosin is the motor protein. It's, as we talked about earlier in the cell, dynein and kinesin were motor proteins. Myosin is the one in muscles, so it goes along microtubule tracks to carry cargo. In the presence of ATP, myosin pulls actin towards the center of the sarcomere. Repeating cycle of attach, pivot, detach, returns, shortens sarcomere, and when stimulation ceases, calcium is pumped back into the sarcoplasmic reticulum, and then the tropomyosin blockade resumes and filaments passively return to relaxed state. So, here's a continuation of the pictures we were looking at earlier. So here's part 5, when the impulse stops. Oops, I don't want to start with part 5, right? I guess this is where we're starting from, sorry. Calcium ions bind to troponin. So troponin is surrounding this thin filament. The troponin changes shape, moving tropomyosin on the actin to expose active sites. So it just changes the conformation to expose the active site. So it changes the shape, so the active site is open for binding. And then myosin heads of the thick filaments attaches to the exposed active sites to form cross bridges. Myosin heads are also important in a function that we call rigor mortis, which we'll talk about later. So number 4, myosin heads pivot, moving the filaments toward the sarcomere. So right, these thin filaments, the thick filaments don't move, remember? So the thin filaments are moved in toward the sarcomere. ATP binds myosin heads and is broken down into ADP and PI. So this is active transport or active. It uses an ATP, so it needs energy. Hence, there's why there are so many more mitochondria in muscle cells than any other cell. So myosin heads detach from the filaments and return to their previous position. The repeating cycle of attach, pivot, detach, return, slide thick and thin filaments past one another. The sarcomere shortens and the muscle contracts. The cycle continues as long as calcium ions remain bound to troponin. So when the calcium, because remember, calcium is released in the contraction of muscles, so it's released into the environment and it can bind to these filaments. But when the calcium is reabsorbed into the sarcomere, this is when it goes to a relaxed or non-contract stage. So a motor unit is a single motor neuron, and the muscle fibers it controls. So it's one neuron per muscle fiber, right? So a single motor neuron and the muscle fibers it controls is a motor unit. A motor unit contains only some of the muscle fibers in an entire muscle. When a motor unit is stimulated, all muscle fibers within it contract, and the size of the motor unit and degree of control provided are inversely related. So small motor units provide precise control and vice versa. Eye muscles have small units, and lower limb muscles have large units, right? So the tinier the muscles that you have, the more control. So it's like in our hands too. We have these really small muscles for coordination of each finger. And in our feet, we have muscles too, but we cannot, as we can use our carpals and metatarsals in our hand, there's not as many muscles in the foot to move our tarsals and metatarsals. So that prevents so there's more larger muscle, which means it's less efficient, and coordinated movement is not as well defined. Movements that require more force recruit more motor units. So when one motor unit is at its capacity, it will ask neighboring motor units to come on board with it to try and lift something up. So for example, when you're trying to lift up, say, a weight, and you lift it up, and you're able to grab onto it and hold it, but then when you actually try and lift it, other muscles start getting involved. So you can lift it all the way up off the ground. That's an example of this motor unit being stimulated and all the fibers within it contract, and then recruitment. So recruitment is very important for muscle fibers. So every muscle employs recruitment. So here's the motor unit, right? So here's the fascicles of muscle fibers, the neuromuscular junctions. So the red, right, is going to be arterial, so blood is flowing through them, and venous, so this is blood flowing out of them, is the blue. So muscle tone and contraction types. So muscle tone refers to constant tension. Constant tension in resting muscles. So motor units are stimulated randomly to avoid fatigue. Two types of muscle contraction, and you should know these. Isometric contraction, so the length is constant and the tension is changing, right? So this means that the small filaments aren't contracting because the length is constant, it's the tension is changing. So whatever's on the outside is causing more pressure. Isotonic contraction, tension is constant and the length is changing. So this is where your muscles shorten and lengthen. So muscle shortening is called concentric. Muscle lengthening is called eccentric. And eccentric, like in math, eccentricity means oval-like shape. So you can just think of the muscle moving in an oval-like shape manner. So isometric versus isotonic. So here's isometric, no movement and muscle tension and resistance up here. And then muscle tension and resistance have moved here. See, this is the bicep, but see this bicep is contracting and this movement is going upward. So isometric contraction, muscle tension is less than the resistance. Although tension is generated, the muscle does not shorten and no movement occurs. So it's like holding a heavy object with no intention of lifting it up. Muscle isotonic is muscle tension equals or is greater than the resistance. So the muscle shortens and movement occurs. And this is if you're going to lift that way up towards your chest or lift up in general. That's an isotonic contraction. So the types of skeletal muscle fibers are three.